I would like to start with like to acknowledge and thank the people who've contributed to the work that I'm discussing today. First and foremost, the individuals and families that have been part of our trials and advisory boards. Also our Roche early and late stage teams, as well as external partners. This summary gives you an idea of what I'm going to cover today. The title of the talk is Developing ASD Therapeutics, which is a very broad topic. The way that I thought might be a good way for me to approach this would be to walk you through our Balvactin program, discuss some of the learnings that we've had from our trials, then discuss our thoughts about where we are and how we're moving forward. So I'll start with a little background on ASD to set the stage. Most of you know most of this, but it's sometimes good to be reminded how big this really is. You know, it's 1 to 2% of the world's population, 1 in 54 kids in the US, no current treatments for the core symptoms, significant morbidity, mortality, significant functional impacts and financial impacts. So this slide discusses a little of the preclinical and clinical and early clinical work that we first built our scientific beliefs on that made us feel like we might be on the right track with Balvactin, which is a vasopressin 1 antagonist. It just highlights some of the thoughts behind our preclinical models, some of the links between vasopressin and oxytocin that was thought of at the time. And then following that, we had our vanilla trial, which was our phase 2 and really first significant clinical data that we had. This is the flow of our clinical program, which transitioned from animal models to the first human proof of mechanism study with a tool compound. It was followed by genetic validation studies and work to better understand our epidemiology, as well as a functional MRI study to better understand our mechanism of action. Then at the bottom here, you see our adult and pediatric phase 2 studies. So one of the big challenges of developing therapeutics in ASD is what we call here building the bridge while you're walking on it. So while we're running early clinical programs, we're also working with experts and sites and networks to figure out potential endpoints, biomarkers, and clinical trial infrastructure. For ASD, there really isn't a great playbook for this to date. And as you can see from this slide here, the vision for Balvactin was to be the first pharmacological therapy approved for the treatment of the core symptoms of socialization and communication. We have 2 phase 2 clinical trials, one pediatric, which was aviation, the one farthest to the right here, one adult, which is vanilla, and one phase 3 clinical trial, which is the one in the middle, viaduct. And as you can see, you always wonder how these things get their names. They're all supposed to have V1A in them somewhere. So that was the consistent thing holding them together. So the vanilla trial was our first phase 2 trial. And you can see here from the breakdown, it was a trial to look at the safety and efficacy of Balvactin on social communication interaction. It was only for men, adult men, higher functioning, so IQ greater than 70, a CGI severity score of greater than 4, SRS of greater than 16, and we had 223 patients in that study. The design was US-based, 26 US sites, randomized double-blind phase 2 trial. We had 3 doses. We had a 1.5, 4 milligram, 10 milligram dose and placebo once a day for 12 weeks. So what happened with vanilla? First of all, our primary endpoint, the SRS-2, was negative, but our secondary endpoint, the Vineland-2, looked as if it may be showing some improvement in a dose-dependent manner. And when we plotted it out with our PK/PD data, and I unfortunately didn't include that slide here, it even gave us more evidence that that could be the case. For us at that point in time, we felt that despite the primary endpoint being negative, that there was such a high unmet medical need and it was an evolving field that we would proceed cautiously. And as you know, hope is a powerful motivator for most of us. So we discussed these results with the FDA and got the green light to move forward into the phase 2 pediatric program. Also thinking that if we saw these improvements in our adult trial, then possibly the pediatric response could be different or maybe even better. And I think most of us believe that, in neurodevelopmental conditions, that the earlier you treat, you could have a bigger effect. So this slide gives you a look at some of the differences and consistencies in our 3 different trials. The vanilla, which is what we were just talking about, adult males, 18 to 45, US only. Viaduct, we expanded that. So we went to not only US, but we expanded to Canada and Western Europe. And we had a little bit longer stability period, and we excluded more medications that we felt like we may have seen issues with vanilla. In aviation, we kept it a little bit closer, US only, but again, expanded to females in that population. The consistencies were in our eligibility criteria were IQ still greater than 70, significant impairment, so SRS-2 score greater than 66. I think most of the others are pretty self-explanatory. Okay. So despite trying to keep as much consistency as possible, which is, as you probably know, is very difficult to do, you can see here that there were some differences between our baseline scores in our phase 2 and phase 3 trials. I'll point out specifically the IQ scores. So you can see there's somewhere between a 6 and 10 point difference in the vanilla and viaduct. And again, vanilla phase 2, viaduct phase 3. And from our primary endpoint, somewhere around even up to like a 10-point difference on this score, which is pretty significant and more than we expected. If you look at the results between the vanilla trial and the viaduct trial, you can see again, these are pretty different. The vanilla trials we saw before, we felt like there was a little bit of a dose-dependent looking curve. I know these margins are pretty wide. But for viaduct, we even saw the placebo seeming to respond a little bit better than our active treatment. Now to be fair, when you take out some really extreme scores, so scores over 30 or 40 on the Vineland, it looks pretty equal. And we'll cover that in just a second. But still different than what we saw with vanilla. We did go back and look at how some of these baseline differences could be impacting the results. And here's one of the examples of some difference. This slide is comparing subjects with a baseline Vineland 2-domain composite. So again, the 2-domain composite, I may not have mentioned this, is the social and communication domain. So it's just leaving out the activities of daily living component of the Vineland. So this is above and below 60 on that 2-domain composite. Subjects with lower scores had a much more of an overall change, and in this case, the placebo change was quite large. You can see the comparison here for these patients under 60, specifically on the placebo change from baseline, which is the second red box. At 12 weeks, 15.7, and 24 weeks, 17.0, compared to the column all the way farthest to the right, which you'll see a 1.1 point change and a 3.2. So pretty significant in that placebo group and pretty significant across the board. The phase 2 pediatric trial also read out negative, unfortunately. But again, the results look very different than our other phase 2 study and even the phase 3 study that we had. So as you can imagine, we spent a lot of time over the last 6 to 8 months thinking about why we had such different results in these 3 different trials. We've looked at a lot of different factors, including age and sex and IQ, geography, some of the baseline scores, which I showed before, placebo response. This was something that was very significant in our study, much higher than what we thought it would be. We tried to look at different things that could be playing into that, but even despite that, we don't think that this placebo response was the only reason that our studies were reading out negative. We also saw that there was a difference in site type, and level of experience, which I'll cover a little bit in a second. Another breakdown in site type that we have that I'll mention is the academic versus commercial sites. Early on in our phase 2 study, we had a much higher proportion of academic sites versus the phase 3 trial. And the reason behind this is that, as you may know, the academic sites tend to get up and going much slower than the commercial sites. The commercial sites, for the most part, use the centralized IRB, whereas the academic sites a lot of times will have their own IRB, and contracting often is more difficult and time consuming. Academic sites have many studies going on, and priorities may not be the same in between these two different site types. So anyway, differences there we'll talk about in a second. And overall, the commercial sites, we saw a pretty significant difference in the placebo rates. But let's start here with rater effects. And what we always knew, that rater quality and performance was going to be really important for our trials. We put several quality control measures to try to keep our raters performing well and consistent. We had a lot of things in place that monitored how these raters were performing. We actually recorded all the scales that they were doing. We had customized programs that were tailored to specific raters and what we felt like they might need. And in spite of all of this, we really weren't able to balance all this out. And it really wasn't even specific to the inexperienced sites. It was just a lot of rater inconsistency across the trial. One of the things I think, and again, I mentioned this briefly before, if we remove all the huge swings that we saw in the Violin-2, the Viaduct study looks like that placebo and active are pretty equally in comparison, as compared to looking like the placebo outperforms the active now. So those are just a few outliers that seem to move that curve. We set up what we called a rater academy to help train raters that may need some more training. On the right here, placebo effect, again, something that we considered highly. We did additional trainings on this at all the investigator meetings. We trained on this. We had specific trainings at all the sites for this. And the quality control, as I mentioned before, we recorded all the administrations of all the trials. And these recordings were reviewed by our CRO, and they triggered extra training for raters that they felt like needed it. If you look at our results from different site types, which is really interesting, we thought. We broke sites into two broad categories, academic or commercial. For the most part, it was pretty simple, but there were some sites that were a little of both. So we had to best decide which they were more like. But the important thing here is that you can see the placebo rates at what we call the commercial, or consider the commercial sites, were much, much higher than what we saw at the academic sites. You can see even 12, 24-week differences, but on the left here, the academic sites, on the right, the commercial sites, which you had placebo rates being much higher and even much higher than the active arm. Another interesting finding was where the patients came from. So if the patients were known to the sites, then placebo rates were lower compared to subjects that were not known to the sites and came in as a referral from advertisements, for instance. Yeah, pretty significant. And what's interesting, we've seen this across not only in neurodevelopment, so in neurodegeneration, in our Alzheimer's trials, we've seen a very similar profile. And of course, this makes some intuitive sense, but you wouldn't guess it would be as large as it is, and the placebo effect being this much bigger. So what have we learned, and how are we moving forward? We've learned a lot from our trials for sure. The rater trainings that we were provided were not enough to level the playing field. There were some sites that just seemed to perform better and more consistently. We're looking at ways to capture what some of these sites did really well and figure out ways to share it with other sites. With endpoints, I think we all can agree that we need to find better ones. The last box here, but certainly not the least, and maybe the most important, is we've been painfully reminded again how heterogeneous the ASD population is, and how we need to continue to develop ways to select the right subsets of individuals for our treatments. Excuse me. So this slide demonstrates what we might be seeing in our studies. It is possible that the vanilla study could just be a different, in some ways, like what we saw in the baseline data. It is possible that vanilla just happened to sample a group that responded better. So this was an attempt to try to demonstrate that. Now, this slide gives a peek at some data that we're working on and not quite ready to share. So that's why I've had to remove the identifiers on the X and Y axes. But we do have some preliminary data here that we're currently running a confirmatory study on and looking to see if some genetic differences that we saw within our study could explain some of the reasons why we think we might have seen something in vanilla. So hopefully more on this to come. Another part of our program that I'm really excited about and hopeful about is our digital biomarker program. One of my colleagues is presenting more on our biomarker approach next Sunday, so next week at this same symposium. So maybe you can check that out. We're trying to use a mix of minimally invasive active measures along with passive measures here to get lots of data on our patients to get better and more objective picture potential change over time in our trials. And one thing that's interesting, I'm sure Chris will talk about next week, is just the amount of data that you get with these digital biomarker trials. It's just massive. And going through it, we got the futility analysis for the Viaduct study, which is where we had this first digital biomarker sub-study, and they're still trying figure out the best ways to parse out these massive data sets. And the last thing I want to mention is our digital therapy program, which is one of the things we've noticed in our trials, both adult and pediatric trials, is that how few subjects are receiving any types of behavioral therapy at all. And we all know how inconsistent that therapy is, even if they are receiving it. And wouldn't it be nice if we could standardize that therapy in our clinical trials? That is one of the goals of this program. And I hope that you'll be hearing more about this over the next year or so. So with that, I will end and highlight a few key messages. And I think we all know these treatments. There's a lot of treatments still desired to treat the core symptoms in autism. Heterogeneity remains a key challenge. And I think one of the more important things to highlight here is the partnerships. Really what we need to be able to move this forward are finding better ways to work together and motivating young and talented people to continue in this field and to accelerate this kind of work. So I'll end on that. Thank you.
Thank you, Dr. Saunders, for that. Really nice overview of, wow, the Balvactin program is pretty complex in terms of all the different things that went into considering how to construct those trials and all the measures that you went through to try to normalize things across sites. And I think it just highlights how challenging that really is. And so I've one question that it's more of I don't know if it's a philosophical one or to me it's practical, but is yeah, I can see now this sort of tension between getting the right sites, which is going to take longer, and getting the study done quickly. And I think what we see here is an example of, it sounds to me like what you were indicating is that the academic sites just generally are better prepared to administer these complex instruments, despite all the training you might do. So I'm at a small company, right? How would you recommend or advise a small company to balance those tensions?
Yeah, no, this is a really good question. And two, when you do phase 3 trials, you're supposed to be representative of the more general population, right?
Sure.
So, that's one of our questions too. We have other programs going forward. We have another ASD asset going into phase 2 right now. It's a GABA molecule.
Mm-hmm.
We're going to the FDA to talk about how to structure those trials. And I think that it does highlight just how delicate or fragile, I guess, this system is. Our hope was that if we did the right thing with our training, maybe we could overcome some of these potential differences between the sites. But
Mm-hmm.
Again, I think it's pretty clear that that's really difficult to do. I think moving forward, we're going to be much more aware of this. I think we will take the extra time, if possible, to make sure that we have the right sites. And again, where you draw that line is really the tough question.
Yeah. Mm-hmm. Yeah, and I think, I think that line actually may be drawn in a different place depending on what the instruments are you're using as well, right?
Yes, exactly.
So, things like ABC, for example, where it's been around a lot longer and it's a much more straightforward instrument, maybe you can, balance it a little differently. But
yes. No, it's a very good point. Yeah, each of these scales are different, and the Vineland is complex to say the least. So,
we do have a couple of questions now from the viewers. One is, did you test any correlation with the subject's baseline vasopressin levels? I don't even know if you can do that. So,
yeah, that's a really good question, and we actually did not do that. We had thought about that. I think in some other studies, for instance, like Lynn Sikage's oxytocin study, they've done similar things. But no, we did not test baseline oxytocin levels. I mean, there's some question on how reliable those levels are. They change even throughout the day. So we really couldn't figure out a good way to do that. But it is something we thought about. It was just, we never figured out the best way and really a reliable way to do it.
And is that partly because what you might measure in the blood at a certain time may not reflect what's actually in the brain, or is there any known relationship there?
Yeah, that's part of it. I think even the blood levels change, and then how that correlates with, CSF levels. Again, like I don't think we ever got to the point where we felt like that enough was known about that for it to be that useful.
Okay. Thank you. And another question, do you think additional biomarkers will help subtype and screen patients further for follow-up trials? And I guess the leading question there is what might those be?
Yeah, no, yes, for sure. And, I think some of the things that we're really excited about are EEG combined with machine learning. I think there have been a few papers published, just in the last 6 months or a year that have been really exciting in this area. And then metabolomics, I think, are another approach that we feel like could have some potential. And then, of course, genetics. So those are the 3 areas that we're focusing the most on, and trying to figure out, if it's one, or if it's if there are ways to even, combine these multiple dimensions for assessment.